All right, I'm gonna do a little more drawing for you. This time it's gonna be really drawing. It's gonna be a cartoon or a simplified drawing of the DNA replication process. In, I'll just say in E. coli, and many other prokaryotes are very much like this when they have a single circular chromosome. But just for specificity's sake, I can only guarantee you that this is what happens for E. coli. Okay, so when we start off, we have one big loop of DNA. That's supposed to be a circle. That's not anything. That's just a mistake there. So imagine, if you will, a perfect circle of DNA, or a perfect loop. It's never going to show up as a circle like this because it's going to be all packed away. But we've got this circles are going to represent our ORI-C site, our origin of replication, and this box down here is going to represent our TER site, where we're going to complete the replication to form this the two replicons. Okay, so DNA copying, the replisome is the DNA replication machine that is going to make our two copies of the DNA. So now what I'm showing is replication going from the ORI site. So now we've got two copies of the ORI site. Two copies of ORI site. And replication is proceeding in the direction of the arrows. Into the fork, as it is known, the replication fork. And at this point, this is what we call the theta structure, because it looks under the electron microscope like the Greek letter theta, which, if you didn't know, looks like that, kind of like the letter O wearing a belt. That's, again, not supposed to be there. Just gonna fill it in. There we go. Letter theta. So the Greek letter theta, it's thought that that's what this structure looks like. Therefore, theta structure. A lot of Greek being added into scientific jargon, as we've seen with things like our alpha, epsilon, and theta subunits of DNA polymerase 3, gamma, phosphate in ATP being cleaved off, things like that. Okay, so now you can see we've continued our replication as we approach the TER site. Maybe these two replicons separate cleanly, but there's a couple of problems that may occur. One is we may get what are known as catenanes. And catenanes are when we have two, the two replisomes are linked to each other, like a pair of linking rings, like two links in a chain are attached to each other. Well, we don't want two links in a chain. We want two separated links. So in order to do that, we have to resolve those catenanes with, this is where topoisomerase 4 comes into play. Remember, that's part of our replisome, is having topoisomerase 4. In case we get into this problem, then we can end up with having two discrete replisomes with their own origin and terminator sites go in their separate directions. So it's going to break these links and anneal them back together. So we've got two autonomous copies of the genome. Another thing that can happen is dimerization. And when dimerization happens, we end up with two copies of the replisome, the replicon that are connected such as like this. So we've got our two origins and two terminator sites, but we've ended up making one big loop of DNA with two copies of the genome in it. And so how do we get those apart? We're going to need to use another enzyme, XRCD, which is going to resolve the dimers. Okay, so this is just a very crude sketch of some things that happen as DNA is replicated in E. coli. So recognizing we've got an ORI site, we've got a terminator site, the theta structure forms, and once we're getting done with completion, we've got a couple of problems that may arise, and we've got solutions to those problems. Okay, see you next time.